and then only you will understand the part 4 video. So let us study light reaction in photosynthesis. So in the last class we have seen when light falls on the photosystem, both the photosystem PS2 and PS1, it becomes active. The wavelength of the light should be less than 680 nanometer, then only PS2 will function. If this is PS2 and PS1, both will become active, and the electron, it will excite electron. This electron, when it comes back, it releases energy. So already this has absorbed, PS1 and PS2 has absorbed, PS2 and PS1, it has absorbed light energy, and the electron is excited, and the electron when comes to the ground state, it releases energy. Now this electron in both the system, it is accepted by the primary electron acceptor. Okay, so in PS2, when the electron it becomes excited and comes back, it is accepted by primary electron acceptor, and energy is also released, and this energy is also passed on. And from primary electron acceptor, the electron is given to ETC. What is ETC? Electron transport chain. Electron transport chain consists of 3 electron carriers, which we have seen in the last class. From the electron transport chain, it is always the electron moves in, the energy is downhill. Why is the energy downhill? Because whenever the electron passes from one carrier to another electron carrier, so the energy always decreases, or energy is reduced. And whenever the energy passes from one pigment to another, when electron passes from one pigment to another, energy always decreases. Here from electron transport chain, the electron moves to photosystem 1. The electron, it moves to photosystem 1. And in the primary electron acceptor of photosystem 1 is iron sulfide. The primary electron acceptor of photosystem 1 is iron sulfide. The electron is given to iron sulfide. From iron sulfide, the electron, it is given to a protein called ferrodexin. This is the protein called ferrodexin. This protein, it gives the electron to NADP+. The protein, it gives ferrodexin, it gives the electron to NADP+. This is electron deficient, and here reduction takes place, and NADPH is formed. The formation of NADPH, it takes place in the stroma, but all this reaction, it is carried forward in the thylakoid membrane. Only the formation of NADPH, it takes place in the stroma of the chloroplast, and rest of the reactions, they are taking place in the membrane of the thylakoid membrane. In the thylakoid membrane. So of the granule thylakoid. So once more we will start. When light falls on the chloroplast, the photosystems become active. Light should be less than 680 nanometer wavelength. Both the photosystems, they become active. If this is photosystem 2 and this is photosystem 1, both will become active when the wavelength of the light is less than 680 nanometer. Both will excite an electron. The electron which is excited by photosystem 2, it comes to the ground state, and it is accepted by the primary electron acceptor of photosystem 2, and the primary electron acceptor of photosystem 2 is pheophytin. Then the electron, it is given to electron transport chain. From here the electron, there are 3 electron carriers here, and from here the electron, it moves to PS1, photosystem 1. Here the electron, it is accepted by iron sulfide. Then again the electron is given to a protein called ferrodexin, and this electron, it is given to NADP+, which is in the stroma, and NADP+, it undergoes reduction reaction, and it forms NADPH. Because it accepts electron and it undergoes reduction reaction, it gets reduced and it forms NADPH. So this is how NADPH is made in the stroma. This NADP, the reaction is NADP plus proton plus electron gives NADPH. NADP plus hydrogen ion, proton plus electron is NADPH. Now NADP plus electron is given to NADP plus, which is already electron deficient. So 2 hydrogen ions and 2 electrons are added, and thus it makes NADPH plus H plus. So the electron is given to NADP plus which is present in the stroma, it is already electron deficient. So two protons and 2 electrons are added, and it takes 2 electrons and NADP plus, it becomes NADPH plus H plus. So the full form of NADPH is nicotine amide adenine dinucleotide phosphate. Full form of NADPH is nicotine amide adenine dinucleotide phosphate. Now PS2 has given its electron and it has passed to PS1, and already NADPH is made. NADPH is synthesized in the stroma by the reduction reaction. Now PS2 it becomes electron deficient. PS2 it becomes electron deficient, it has given its electron. Now however light falls, PS2 will not function. It is electron deficient. It will not excite electron because it has no electrons. Now in the presence of sunlight, PS2 it has enzymes which undergo and it carries out photolysis. In the presence of sunlight, PS2 it has enzymes which will carry out photolysis. Photolysis is breaking of water to produce oxygen, and this reaction it is occurring in the lumen of the granule thylakoid, and lumen, that is empty space of the granule thylakoid. Now PS2 it has no electron, it is electron deficient. However light falls, this PS2 it will not excite electron. What will happen in the presence of sunlight? PS2 has enzymes which will carry out photolysis. Photolysis is breaking of water, and water will break, it will give oxygen, electrons and protons, and this electrons will be taken by PS2. Photolysis. So already we have seen when photosystem 2 it becomes electron deficient after giving its electron to photosystem 1. So photosystem 2, however light falls on photosystem 2, it will not excite electron because it has no electrons. So photosystem 2 which will carry out photolysis. Photolysis is breaking of water, and this happens in the lumen of the thylakoid membrane, lumen of the granule thylakoid. Lumen is the empty space. So breaking of water, this photosystem 2 it has enzymes. Photosystem 2 has enzymes which carries out breaking of water, and breaking of water gives 2 protons, half O2 plus 2 electrons. H2O gives 2H plus half O2 plus 2 electrons. These electrons they are accepted by photosystem 2. So this process, breaking of water is an oxidation reaction. Breaking of water, it is an oxidation reaction, and the electrons it is accepted by photosystem 2. Manganese and chloride ions, Mn plus Cl minus, are also involved in photolysis. They also help in the breaking of water. So photolysis happens in the lumen of granule thylakoid. When the photosystem 2 has no more electrons, it has lost electrons, it will not excite its electron however light falls on it, and it will carry out photolysis. Photolysis is the breaking of water which happens in the lumen of the granule thylakoid, and breaking of water is an oxidation reaction. It involves manganese and chloride ions. Photosystem 2 has enzymes which breaks water to give 2H plus, half oxygen plus 2 electrons. These 2 electrons are accepted by photosystem 2. Now the main function of light reaction is synthesis of NADPH, which we have already seen. Synthesis happens in the stroma, and second one is making of oxygen, and oxygen is made in the lumen of granule thylakoid, and third one is synthesis of ATP. So in the light reaction, 3 things are synthesized: NADPH, oxygen and ATP. Let us see the non-cyclic flow of electrons in light reaction. Non-cyclic is it is not cyclic, it is not cycling. When light falls on PS2 and PS1, both of them they excite electron. This electron will never return back to the same photosystem. For example, when the electron is excited by PS2, the same electron will not return. The electron it will not return to PS2. The electron is not given back to PS2. We have seen it will pass on to primary electron acceptor, and finally it will goes to PS1. So the electron which is excited by PS2, it never returns to PS1. So this is non-cyclic flow of electrons. Similarly, the electron which is excited by PS1 will not return to PS1, and it is utilized, it is given to the primary electron acceptor and so on. So in the non-cyclic flow of electrons, three products are formed: oxygen, NADPH and ATP. ATP, how ATP is formed? ADP plus phosphate is ATP. On adding a phosphate to ADP, we get ATP. Three products are formed, but the electron, it will not follow a cyclic pattern. The electron which is excited by PS2 will not return back to PS2, it will go to PS1. Similarly, the electron which is excited by PS1 will not return back to PS1 and it will go to NADP plus to make NADPH. So this non-cyclic flow of electrons, it forms, this is called as non-cyclic photophosphorylation. In the non-cyclic photophosphorylation, what and all is made? Oxygen, NADPH and ATP, and the electron does not follow a cyclic pattern. Now, there is another process called cyclic flow of electrons, which is called as cyclic flow of electrons, in which the electrons, they form a cyclic pattern. And this is known as cyclic photophosphorylation. It is also known as cyclic photophosphorylation, in which photophosphorylation is in the presence of light, ATP is made. So in cyclic flow of electrons, if light is given, light is having wavelength which is more than 680 nanometer, PS2 will be non-functional, but PS1 will become active. When light has wavelength more than 680 nanometer, PS2 will be not active, but PS1, it will become active and it will release an electron. So friends, in cyclic photophosphorylation or cyclic flow of electrons, what happens is there are two photosystems. When the wavelength of light is more than 680 nanometer, photosystem 2 is non-functional. The electron in the photosystem 1, it becomes functional and it excites an electron. This electron is accepted by the primary electron acceptor, that is FeS, iron sulfide FeS. From here, the electron goes to plastoquinone. From plastoquinone to cytochrome B6F, and from here to plastocyanin, and again the electron returns to PS1. This process, it helps in the formation of ATP. It helps in the synthesis of ATP. So this is known as cyclic flow of electron or cyclic photophosphorylation. In this process, there is no NADPH or oxygen is made, only ATP is synthesized. So in cyclic flow of electrons or cyclic photophosphorylation, the PS2, when the wavelength of light is more than 680 nanometer, PS2, it becomes excited, releases electron, and electron is accepted by primary electron acceptor. Electron goes to electron transport chain, that is plastoquinone. From plastoquinone to cytochrome B6F, B67, and then cytochrome B6F, and from here to plastocyanin, and from here it returns to photosystem 1. So after going through the electron transport chain, it comes back to photosystem 1 and it synthesizes ATP. So this is known as the cyclic flow of electron or cyclic photophosphorylation. Here ATP is synthesized.